Hello, everyone. Good morning, good afternoon, and good evening, and welcome to Jack's Live. I'm your host, Jenny Morganwick, and joining today is Doctors Lori Case and Elena Gonzalo-Gill, both study directors with our in vivo pharmacology group here at the Jackson Laboratory, and they're coming to you live from our Bar Harbor location. So welcome. So today we're going to talk with Lori and Elena about autoimmune disease models and how they can be used in preclinical drug discovery. But before we get started, just a few housekeeping details for you. So during this broadcast, we would love to hear from you. So use the comment box that is under this video, either on LinkedIn or Twitter, wherever you're tuning in from, and submit your questions, your comments, drop us a note, tell us where you're tuning in from, and we'll try to get to as many of your questions throughout the live stream today, but we will have a question and answer session at the end of the interview, so please stick around for that, and we'll try to get to everyone's questions. So hi, Lori. Hi, Elena. How are you guys today?
Hi, Jenny.
Hello.
Doing
How
well.
are you?
Great. So let's dive right in. So to kick things off, I would like if one of you could provide everyone with a high-level overview of disease area platforms and services that someone can use surrounding preclinical research in the autoimmune area.
Sure, I can take this first question, Jenny. While there's more than 80 types of autoimmune diseases that people can develop, the large majority of preclinical research and drug discovery, it's dominated by a few key disease areas. And this is largely driven by how many people have the disease, as well as the availability of relevant mouse models that can be used to facilitate preclinical research and drug discovery. So type 1 diabetes, lupus, and psoriasis, these represent key diseases researchers are studying, and JAX also offers in vivo services in these areas.
All right, so I know that there are some therapeutics that are already approved for the use that span various autoimmune diseases, and they're actually quite effective as treatments. Is there any particular advantage to working with a group that has multiple autoimmune platforms readily available?
So it is true that current therapies are often successful because they can work not only just on one type of autoimmune disease, but they span across several disease areas that share common biology. However, preclinical drug discovery is still very active in these areas due to limitations in the efficacy of current treatments across the patient population. So in other words, what works for one individual might not work for another. And it's for these reasons it's important to consider using multiple platforms and models when testing novel therapeutics.
Yeah, and actually, I think one of the best examples I can think is adalimumab, also known as Humira, because this therapeutic has been approved for treatment of several autoimmune diseases, including rheumatoid arthritis, psoriatic arthritis, Crohn's disease, ulcerative colitis, and plaque psoriasis. So as you can see, it can be quite powerful to test your therapeutic compound across many autoimmune diseases.
Oh, it looks like we actually have our first question from the audience. So one of our audience members would like to know if we offer any other platforms here at JAX. You've already mentioned lupus, type 1 diabetes, and psoriasis. Could one of you comment on that?
Sure. It's actually, as we speak, we're actively validating platforms for multiple sclerosis, and that will model different forms of disease progression. And we're also validating models for rheumatoid arthritis, as well as a spontaneous model of ulcerative colitis.
Oh, so it's great to hear that you've got more platforms in the pipeline, and thank you to our audience member for the question. So since I have two of my study directors at my disposal right now, I would like you guys to maybe tell us what a typical autoimmune study would look like.
Sure. So, a typical study begins with first working with the client to understand their goals, and then developing a study that is designed to meet these goals. So this includes identifying first the optimal mouse model for testing based off of the mode of action of the test article, and including relevant clinical readouts during the study. So for example, if a client is testing a therapy that protects against the production of autoantibodies, we would recommend using the MRL-LPR mouse model of lupus. These guys produce very high titers of antibodies that can be measured in the serum throughout the in-life phase of the study. And in addition, we could include urine ACR measurements to evaluate kidney damage or evaluate peripheral blood cells and flow cytometry in order to track efficacy of the test article.
Yeah, and Lori, I think this is a good time to also mention that studies are entirely customizable to induce the specific readouts of interest. First, I think it's very important for researchers to identify your goal. And what I mean is you first have to identify the target of your therapeutic and which readouts have other researchers used for their studies. Of course, it's important to try to replicate what they have published, but also think what you can do to differentiate from other researchers. For example, like a novel technique that can be applied for your study. Also, something important to keep in mind is that most of these platforms have a prophylactic and therapeutic design, meaning your treatment before or after onset of the disease. And as a researcher, you should definitely take this into consideration when you are designing your study.
So it sounds like there's a lot of moving parts to a study. So thank you for going through that for everyone. And I am getting a couple people just saying hi from around the world, actually. So just want to remind everyone to remember to post your comments, your questions in the question in the boxes that you see below this broadcast on LinkedIn or Twitter, and we'll be sure to get to them. So I know you guys both have a lot of experience in running many different studies, especially, in the autoimmune area. So I would love if each of you could maybe tell our audience what is, in your mind, the biggest roadblock that a researcher might face when using mouse models for autoimmune disease.
Well, as most of you know, the autoimmune diseases are very highly influenced by the environmental conditions. And those are factors that are very hard to control. So here at JAX, we work with experienced in vivo technicians and researchers because they really understand how important it is to be precise in the technique to try to minimize other factors that can negatively influence in your study. And I think this is something critical.
Yeah, I agree. And along these same lines, roadblocks can emerge just simply due to a researcher's inexperience in running mouse models of autoimmune disease. And because of our experience in working with these models, we've generated reference data on numerous clinically relevant phenotypes used to determine, let's say, for example, the time of dosing start or the duration of dosing. Very often there is significant individual variation within a mouse strain. So for instance, not all NZBWF1 mice, which is a model for lupus, will develop evidence of kidney disease at the same time. And this makes it difficult to determine the optimal time to start dosing for a therapeutic study. To circumvent this, though, we pre-screen mice for markers of kidney damage and use a rolling enrollment approach to only start dosing animals when they meet critical established criteria.
So we've talked about a couple of roadblocks. How about if both of you could share some words of wisdom to anyone thinking about next steps for preclinical evaluation for their drugs?
Sure. And, we've already touched on this, but I think it's worth reiterating the importance of choosing the right mouse model for the mode of action of the drug being tested. So, for example, there have been instances when a client asked for a study design using a streptozotocin-inducible model of type 1 diabetes, and they want to evaluate the efficacy of their immunomodulatory drug. However, type 1 diabetes in this model is not the result of an autoimmune response, rather it's chemically induced. So the use of this model would likely yield negative data for the client. Instead, the NOD model is the quintessential autoimmune model for this disease. So we work with our clients beginning at the study design phase, and we can often identify potential feasibility issues and suggest viable alternatives just to help ensure optimal study conditions.
Yeah, from my side, I would always recommend you to work with someone or a group that work with a real solid program, and especially that has high integrity and, of course, cares about the science.
So thank you so much. I think everyone can see the value in getting words of wisdom from you guys, since you're so experienced with running a lot of these studies. So that was basically all the questions I have for our interview portion, but I wanted to make sure that we left enough time to get to some of the questions that have been submitted from our audience. So if you guys are up for it, I've got a few questions to send your way. So the first question, I'll let you guys fight out who wants to take this answer. You mentioned investigating potential therapeutics. Can you also use mouse models to study disease etiology?
Sure, I can take this one, Elena. So absolutely, investigating basic biology in animal models has been instrumental in our understanding of disease etiology. Now, JAX is dedicated to discovering the genetic basis of diseases, and we offer cutting-edge animal models to do so, and this includes the collaborative cross mice and the diversity outbred mice. These strains are specifically designed to model the complexity of the human genome, and we can support studies in diseases with complex etiologies, just like autoimmune disease.
Okay. We have our next question is more of a it's kind of a comment and a question combo. So the comment in question are mouse models offer several advantages to studying a wide range of diseases, but how close are these models to what is observed in the human population?
Yeah, I think I can answer this question. Of course, we all know that mice aren't humans and humans aren't mice. And although some clinical trials have failed after generating successful data in mouse models, the use of mouse models have been extremely important for the identification of efficacious therapeutics in autoimmune diseases. Some examples from lupus include some antimalarial treatments like hydroxychloroquine, or some other treatments like methotrexate, cyclophosphamide, or belimumab. And the success of this translational relevance relies partly on mouse selection based on the mechanism of action of your therapeutic.
Okay, thank you, Elena.
Sure.
Another question we have from the audience, I think, Lori, I might direct this one to you. Can you describe the differences between the lupus models available?
Sure. So there's a number of different models that are out there. Two of the primary ones that researchers are using are called NZBWF1 and an MRL-LPR model. The biggest difference between them is that the NZBWF1, this model more recapitulates the genetic underpinnings of what we see in the human disease. This model has been incredibly important in mapping gene loci responsible for disease susceptibility. And then with the MRL-LPR model, it's a little bit more contrived. It has a mutation in a gene that causes this massive lymphoproliferation. It's not necessarily a gene that humans develop, but what both models are really great for are that they develop very robust disease. They both will develop very significant kidney damage, lupus nephritis. And, they both have the very clinically relevant hallmarks of the disease, like double-stranded DNA antibody production, urine ACR measurements, a lot of different things that we can measure in life to really evaluate the disease progression in response to a therapy. So those are the two main models. There's a couple others out there that we hope to bring online soon. One would be the BXSBYAA model, another one being a TREX1 model, and we're actively working and hoping to get those online soon too.
Thank you, Laurie. So I think we have time for one more question. I think this is an important one. It's not exactly related to autoimmune research, but it's definitely relevant, and I think you guys can answer it. How is COVID-19 affecting you at JAX?
Yeah, this is a really important question, and I truly hope everyone watching is doing their part to stay safe and healthy out there. And at JAX, ensuring the health and well-being of our community and the mice is our top priority. And we've enacted our emergency response protocols to ensure continued uninterrupted access to JAX-Mycin services. But if you'd like more details, you can visit our website, jax.org/COVID-19-response to get more details on this.
Thank you, Lori. So it looks like we are just about out of time. I'd like to thank both Lori and Elena. Thank you guys for joining us today. A huge thank you to our audience for tuning in. It's great to hear that there is a clear pathway for those researchers interested in preclinical testing in the autoimmune space, and all. The addition of the new platforms you mentioned sounds really exciting. So thank you for sharing that with everyone. Everyone, if we didn't get to your question, we will be sure to follow up via direct message. So be sure to join us next week for another JAX Live event. And I want everyone to have a good morning, good afternoon, and good night. Thank you all.
Thank you.
Thank you.